Alright, so the next experiment is oxidase production. Okay, so the purpose of this one, it's another physiology experiment in which we're talking about how to classify certain organisms not based off of their cellular or colony morphologies. But the purpose of this experiment is to determine the presence of the cytochrome oxidase, and that's another enzyme that we're going to be talking about. But before we talk about that enzyme, we're going to talk about oxidation reduction. All right, so oxidation reduction is a class of chemical reactions that have to do with the transfer of electrons. All right, and so the general principle is that if you're oxidized, you're losing electrons, and if you are reduced, you're gaining electrons. So it kind of looks like it looks at the charge of the molecule. So this molecule right here has electrons on it, and when it loses those electrons, so here we have electrons moving from being on the orange blob to being on the greenish kind of brown. It's just a really bad color blob, right? So so the electrons go from here over to here. All right, and so because the electrons are lost, this molecule right here was oxidized. All right, and now subsequently, this molecule right here gained the electrons, so it was reduced. So the way I kind of look at it is that the charge was on this molecule is reduced because it gained electrons. So it's a reduction right here. All right, and the easy mnemonic to remember this is Leo the line goes Gur. So if you're losing electrons, like this orange molecule, losing electrons, that means you're oxidized. And if you're gaining electrons, you're being reduced. A lot of other people know like oil rig, and that oil is lost in, or oil rig, so oxidized is lost, reduction is gained. But I don't like that one because you don't know what like the thing you're losing is. You just say oxidized is lost. But Leo the Lion says if you're losing specifically electrons, then you're oxidized, and if you're gaining specifically electrons, you're reduced. And that confused oil rig kind of confused me a couple times, so I find this is a better mnemonic to remember it by. But yeah, oxidation reduction reactions are very important with regard to metabolism. And the reason being is because the electron transport chain is a series of redox reactions. So what happens is you have these electron carriers donate the electrons to electron transport chain. Well, a way of looking at that is that you're losing electrons from this NADH, and this electron transport chain is gaining electrons. So this NADH would then be oxidized because it's losing electrons, and the electron transport chain will be reduced because it's gaining electrons. So what happens is that, yeah, the electrons are lost from NADH and are gained onto this FMN, all right, and then they move throughout this, and it's a series of oxidation reduction reactions because, you know, this one is gaining the electrons here, but it's losing it here. This one is then gaining electrons here, but then losing it here. This protein is then gaining electrons here and then losing it here. These are then gaining it and losing it, 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 up until you have O2. And then O2, we said, was the final electron acceptor. It gains it, and then it doesn't lose it, right? We just now have more water in the cell. So it's a series of redox reactions because each one of these proteins is being oxidized and then subsequently reduced while you're pumping protons across the membrane. So this last protein right here is called cytochrome oxidase. So we talked about last experiment how when oxygen accepts these electrons from this cytochrome oxidase, oxygen accepts it and then it makes water here, and then every now and then it makes hydrogen peroxide, and then hydrogen peroxide is bad, so we got to use catalase. And there's another class of proteins called peroxidases that also detoxify this hydrogen peroxide. But yeah, catalase and peroxidase can then detoxify that. Now, and that's how we test for the presence of hydrogen peroxide. But there's a couple different ways we can get hydrogen peroxide that we didn't talk about. So testing for the presence of cytochrome oxidase, this protein right here, is going to be very important. And it's another way of measuring oxidative phosphorylation with aerobic respiration, rather. So testing for these for this protein is important, and the organisms that have it include aerobes, facultative anaerobes, and microaerophiles. And so if you don't know that, you need to brush up on your terminology there. But cytochrome, what it is, it's actually a cofactor in a protein, cytochrome C, right here. And cytochrome, it's a part of, cytochrome is a molecule that is part of the electron transport of cytochrome C that gets reduced, and then it gets subsequently oxidized. So it gets reduced here when it gains the electrons, and then oxidized when it loses the electrons. So it's oxidized by cytochrome oxidase. So this protein is the one that oxidizes this, so it's called cytochrome oxidase because it's oxidizing this cytochrome. So cytochrome oxidase, and it's kind of confusing, but if you really just look at the electron flow, you can see that it's oxidized here and then reduced here. So a molecule that's very similar to cytochrome, or has similar functionality at least, is called Tetramethyl P-Phenylenediamine dihydrochloride. And I have a picture of it right here, and it makes a lot of sense with its name because it's tetramethyl, so we have 1, 2, 3, 4 methyls. P, they're para. If you've taken organic chemistry, these are para groups. Phenylene, so phenylene, and then diamine, so it's got 2 nitrogens, diamine, and then dihydrochloride. You have 2 HCl's over here. So tetramethyl phenylene diamine dihydrochloride is a good way to remember it for your final. And what it does is that it's similar to cytochrome. So let's take a look at this. So I went the wrong way. Okay, so what we did in this experiment is that we streaked 2 organisms on a plate. And what we did is we added that tetramethyl phenylene dihydrochloride to this plate. And so it's an artificial substrate, so it's like fake cytochrome. And it can be oxidized by cytochrome oxidase. The only thing is you have to be really careful when you do that. So you'll add the reagent and it'll turn blue, but you have to read it immediately after you add the reagent, or what can happen is oxygen from the air can actually oxidize the reagent. So you have to look at it really quickly. But then if it turns blue, that means it's in its oxidized form. So that means the molecule was oxidized, so that means that cytochrome oxidase is present in that molecule. So if it turns blue, cytochrome oxidase is present. If it doesn't turn blue like it did in E. coli over here, then it is not present. So relatively straightforward experiment if you understand oxidation reduction and electron transport chain. If not, you're going to have to take a look at it a little bit more. But the big takeaways here are cytochrome oxidase is a protein that it's in aerobic or aspiring organisms. You can test with this with tetramethyl P-phenylenediamine dihydrochloride. And if it turns blue, then it's positive. If it doesn't change color, it's negative. And if it's positive, it has that cytochrome oxidase. So relatively straightforward experiment. The big thing is, the tricky one here is remembering this name. So you'll have to do that. But if you can think of the structure, then the name comes relatively easily.